Hello!
Hi there.
Welcome to Microbiology Journal Club. We nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology at the University of Chicago, where I was infecting skin I grew with MRSA. Then I was a fact checker for pharmaceutical advertisers, and nowadays I'm the president of Biotech Without Borders, a nonprofit based in NYC dedicated to the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked in bacteria, but I've also worked as a research integrity specialist and currently working as an editor for a scientific journal.
Every week we meet to talk about microbiology, and today it's our news week where we're doing an overview of some of the coolest microbiology papers that we've seen during the week.
And at the end of the month, we'll be doing our deep dive. So if you've got any papers you want us to do a deep dive on, then please message us at microtwjc and let us know if something has caught your fancy.
You can follow along with any of the papers that we discuss in our shared Zotero library, linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And I'm a bit sick this week, so you might want to stand 6 foot away from the camera, from whatever screen you're watching this on. But yeah, boy, do we have a show for you today. We have not one, but two papers with opposing views on the origins of SARS-CoV-2. Yes, it's a science catfight, and it gets wild. Plus, we have a look at how twisty RNA could be a target for drugs, and how COVID-19 can change the brain. But wait, there's more. We've got a new way of making vaccines against the bubonic plague. Smart antibiotics, and even not antibiotics, smart vaccines against the bubonic plague. Sickness brain. And finally, we've got a look at how Pac-Man inspired a new way to study bacteria.
Absolutely. And as always, we are kicking off with our papers about SARS-CoV-2. This week, we have this interesting double feature that you found, Faz, starting with surveillance of SARS-CoV-2 in the environment and animal samples of the Huanan Seafood Market.
Yes, so this is a paper coming from the CCD. So this is the Chinese Center for Disease Control. And what it looks at is the possibility that the Huanan market, a market in Wuhan, that was the location. Well, initially it was thought of as the location as the spillover event, the place where the SARS-CoV-2 virus spread out into the community, and then, but then like kind of things changed, and the Chinese CCD said that actually they don't believe that this was the source of the spillover event.
So one of the reasons why like these markets were called the source of the spillover event is just the previous knowledge of original SARS-CoV, right? Like surveillance of these types of markets at that time had shown that there are certain animals that get trafficked through these spaces that end up having that virus. And I think people generally accept that original SARS-CoV-2 was from spillover events in locations like this. So that was like kind of understandable that people thought it might be also the site of SARS-CoV-2. But it sounds like there's a bit of sounds like there's a bit of a debate now whether or not that's the case.
Yeah. So there is this thought that the basic thing is that the Chinese authorities have kind of like stated that there wasn't any trafficking in the kind of wild animals, that there wasn't any illegal trade in wild animals, because I think ever since the first like SARS-CoV-2 pandemic, which was linked to raccoon dogs and other like exotic animals in these kinds
civet cats.
Yeah, civet cats as well. It's because I think there was, for the original SARS-CoV-2, there's a whole bunch of different animals that were indicated as potential sources, because there was like this there was like an ongoing infection within the animals before it spilled over into humans. So what they did here is they took samples from January 1st, 2020 up till March 22nd, 2020, and they collected a total of 923 environmental samples from all the different locations within and around this market, including 457 animal samples, including things from like stray animals and a wide variety of different species as well. So I think like
they have a good map in this paper. Figure 1 has like a map, I guess, of the the west wing and the east wing of this particular
Yes.
market. There's like all these stalls, presumably. Some have cages. What do they have? Like sewers, right? This
Yeah.
is like this. This is an idea of all the sampling that they did around the market.
Indeed, yeah. So the market is spread into two zones. There's the eastern zone and the western zone. So I believe
Mhm.
that the western zone mostly has seafood and animals, and livestock meat is in the eastern zone. I think
Mm.
that's um, and and they basically say that their results suggest that SARS-CoV-2 might have been circulating in the market, especially around the western zone. But they looked at the correlation of the abundance of SARS-CoV-2 and the abundance of other species, and what they concluded was that they didn't find any, they didn't appear to find any circulation in the animals in this market, um, and that most of the strains they did find were correlated to the human strains of SARS-CoV-2. So I think going by the idea that actually like there might have been infections in this market, but actually they came from humans. Because if it was like a
Mm.
spillover event, what you'd expect is there are lots of animal versions of SARS-CoV-2 circulating in that population of animals before it spilled over into humans. Uh,
Mhm.
there are some caveats with this data because the date these comes from samples taken from January 1st, 2020 to March 2nd, 2020. And that is somewhat outside of the window of when SARS-CoV-2 was first discovered from
Mm.
cases related to this area.
Yeah,
So
that's like a little bit later, right? Like
yeah,
people thought it might have been already established at that time.
yeah, I think it was. I mean, SARS, it
Mhm.
was COVID-19 was discovered in 2019. So
Right, right.
yeah, so having samples from 2020 are not it's not going to capture the earliest phase of the outbreak, and
Mhm.
also I think that there were like efforts take once this was discovered in this market. I think efforts were taken to control the the animals in there to disinfect the place and to
Right.
improve procedures.
Hmm.
Um,
Yeah, so this could have been after like a crackdown already in terms of what animals were going through there.
yeah, that is my thoughts on this. But yeah,
Right.
so so this is the first part, and then like rapidly another group published a paper, and so this is an interesting because the lead author on the so I'm going to show the next paper because this
Hmm.
paper is titled "The Huanan Market Was the Epicenter of the SARS-CoV-2 Emergence," published by Michael Warraby, who was I think a couple of maybe a year or two ago, there a group of people said that who are arguing for more forensic examination of the SARS-CoV-2, specifically because they wanted to look into this lab leak theory. So I believe there's this quote they submitted a letter asking for the WHO to have an inquiry, including concluding that while lab leak was extremely unlikely, they felt that they should take it more seriously. And I feel this is the first paper that was the outcome of this kind of more intense analysis. And this is an interesting paper because uh, I get this feels like a very angry paper to me. I, so like so what they do is they first they try to catalog the infections very early on in the outbreak. Um, but the problem is there isn't very much of that data is held by the Chinese CDC. And
Hmm.
so the way they got that data was from extracting from a WHO pamphlet that was obtained very early on in the outbreak. So let me see if they. So yeah, this is actually a very old school way of trying, well, very obscure way of trying to get data. So let me see if I can pull it up because it is. So let's see if it's in. So what they basically did was they basically got this is an image from the WHO mission report. So this is the had it was just so apparently they weren't able to get the data directly from the Chinese CDC. So they took this data from WHO mission report and then they mapped that onto an actual map to try and find the locations. So there's a lot of like graphical tinkering here because they trying to
Hmm.
extrapolate from this one piece of data where these initial locations were. And and then they also add in data from other locations independently sourced as well to try and get an
But
idea of where the first like cases of the outbreak were.
Yeah. So like this is as opposed to having like an Excel sheet, for example, that like gave people like locations and infectious information associated together. Here it was just like a map that was published by the WHO that now they're going to import into their own map, right? Overlay their areas of interest, plus add any more data that they have onto it. And I mean that method for determining like epidemiological centers is like the beginnings of epidemiology, right? Like that's John Snow or whatever, the cholera pump is like done through that method. So it's not a bad method. It's just like people don't do it anymore because we could just have like Excel sheets. You can map these things out without like replicating just another map.
I mean, ideally you'd get like that data directly from the source, which would have addresses and case and more integrated case data. But they clearly didn't have this access to this information, so they had to go through this way to try and get an idea of what was going on.
Just the
so they had to extract the case locations from here in order to try and figure out. And then they did some analyses on this data to try and figure out where the center of it was. So so I mean these kind of geospatial analyses have been, I mean, my most favorite ones for uses of these is they've been used to track down serial killers. Just by but yeah, I think the John Snow example is probably the more apt one here.
And well, that's the epidemiology one, but you know the technology maybe, right? Because like I see now that they're using funny algorithms in order to do this, like that's being enabled by our understanding of computer technology.
Completely. So what they've done is, so they mapped the data out. It's not perfect because again they're trying to extract this from from the CDC, from the data already provided by the CCDC, but filtered through that WHO report.
Right.
And based on that, they tested the hypothesis of whether could this pattern of cases be due to it circulating in the community. So they identified areas of like high population and places where there's lots of elderly people there, and looked at that, because if it was circulating in the community, you'd expect COVID-19 to be hugging population centers and to people who are older, because those people would be more vulnerable. And what they found was that it didn't seem to follow that pattern. What they found was that it tended to center around that Huanan market,
right?
Which seems to indicate that was the center of this outbreak.
But so this is like a really different methodological approach, right? That then is being used to argue that it is the center, versus the approach that was done previously, sampling the animals, right, to argue that it wasn't.
But this is just the beginning of their study, because then they did go into that question of the sampling of the animals, and this is where things get really catty, because so actually
because they also have maps.
They also have timing as well. So they also noticed that the earlier the cases were, the more centered around the Huanan market they were, which is, again, it adds to their thought that this case emerged from that area. But obviously they've got this other paper that is telling a very different story, that actually from the CCDC's perspective, they want to say that it was outside in the community prior to this. So but yeah,
I'm saying that like in the next section, like they actually have maps of the market itself again, and this time they're highlighting I guess different cages, different locations, specific locations.
So what they did was they went through different. So I think one of the things they did was they went through different places to look at the different shops there were, and they also looked at various reports of animals being sold in the market to try and check to see what. So rather than going through so I think so they looked at receipts, I think, and also fines. So they looked at like places that have been fined for selling animals illegally, and they
They kind of did tried to do a historical reconstruction based on the financial, right? Based on like data that they were able to access, right? Historical transactions, and rebuild what they thought the market was like at that point.
Yeah, and
before the pandemic.
yeah, what they've kind of found were reports of that actually some animals that like would be susceptible to SARS-CoV-2 were being sold there that weren't well, they weren't mentioned in other data, and they also have this, again, really unusual for scientific paper. They've got these pictures from a social media user who toured around the markets to raise awareness that all this illegal animal sales were happening. And this user published these articles on the Chinese social media net, and then they were taken down very quickly, and they were received via a journalist. And I think and I think one of these pictures is from one of the researchers themselves 5 years ago. The point of these is to demonstrate that there were illegal animal sales happening, and that it is unusual that they weren't described, that they weren't present in any of the official data. And
so then they come to the opposite conclusion.
Yeah, they come to the conclusion. Well, and they do point out that all of the data reported are for animals that aren't susceptible to SARS-CoV-2, and they kind of plant this implication that there is data being hidden away from us. And I think a lot of this paper is providing that evidence, which leaves us with this kind of real. It seems like a kind of a dichotomy, because what they're doing is they're taking data from like the Chinese CCDC to prove that the Huanan market was the center of the outbreak, and also providing evidence that the CCDC weren't providing all the information we needed. So on the one hand, it seems like, oh, we're trusting the source, on the other hand, we're not trusting the source. But I think from the outside that might seem disingenuous. But I think what they're doing here is that what they're saying is, we know you're doing a cover-up, and we're using your own data to prove it.
Well, I also think that what you said earlier, just that the sampling of the first paper is limited, right? Like they're only sampling from a very specific time point. But here, there's less certainty because you're doing this reconstruction of figuring out what was back then, right, in the market, and not like they didn't have eyes on the ground at that exact moment to sample those animals, but nobody did, it seems like. Or if the Chinese government did, then they're not showing that in the previous paper. So this is the best view that we have into some potential initial events, or like, I guess, initial surveillance that could have been done in this particular area. And really the case here isn't about the animals that are going through. Like, I think that the strongest case to say that there is that things focus is that analysis of overlaying the map data, right? And just seeing, like, based on the timing of the cases, where were the early cases clustered versus later cases.
Yeah. So I think that this is from like a scientific perspective, but also from like the context of this fight that's going on.
Yeah, I mean, the fact that it integrates journalistic sources into it, and right, there's a lot of work that's being done almost at a forensic level in this paper, is not something, I guess, that we would typically associate with doing like viral surveillance, but I guess it's become like a contentious point to speak about, and so scientists want to weigh in as well.
I really think this will have a lot of trouble going through peer review because of that, because I think scientists aren't used to integrating this kind of information, and it's just going to it's either going to be cut down, or it's just a very
right. The claims will have to be constrained a bit more, because here it's like almost like an investigative expose style of publication.
So I think that there is some merit to what they're saying, but the problem is they've highlighted the problem is that it's going to be very difficult to figure out the origins of SARS-CoV-2 when there is a whole lot of data that isn't there. And they've highlighted this, and we don't know whether that data was taken or not. We don't know whether Like, because if you're trying to like run an illegal like wildlife trading, then are you going to let someone to swab your animals? I don't think you would. So we might not ever have important data about SARS-CoV-2.
Yeah, I guess I don't want to say that scientists don't do those types of studies, though, right? But like they would have just been undertaken like in a more controlled fashion potentially, and then also like this is in response to a specific event, right? Like people want to tie like the specific event of the SARS-CoV-2 pandemic to some point in time, and that's going to be a lot more difficult than saying like, does illegal trade happen with animals in this thing, and can that like potentially produce the type of environment that would lead to spillover events? Like those studies can be done. It's just like you can't do them retrospectively onto this past event.
Yeah, it's very difficult because no one knew it was SARS-CoV-2 when it was happening, and that because of that, a
Right.
lot of things wouldn't have been collected. And it's really frustrating to have this kind of because there is so much data here, but it is just yeah, it's so tantalizing. I imagine
I
people.
also want to say like
Sorry.
from the example of like the first SARS pandemic, like that first SARS pandemic, I believe that there were policy things that like that were presumably like people were trying to implement right around these markets. Like that's why certain animals are illegal now to trade in these zones, right? There is an attempt to clean things up, but the world is a big place and it's not easy to do 100% of anything. So like, I think that and also like there's also the priorities, right? Like sometimes like not enough funding, not enough eyes on a specific problem. Like those things can lead to basically lapses in regulation that could result in the spillover event.
Yeah, it's the and the next paper is related to these, but it's trying to like kind of square the circle on a on a data point they found where that they found that so there are two lineages of SARS-CoV-2, lineage A and lineage B, and what they're expecting to find maybe one lineage appearing in, but then they found like lineage A like showed up like much both lineages showed up at the same time, and they tried
Mm-hmm.
to figure out what the cause of that was. So that led to this paper called SARS-CoV-2 Emergence very likely resulted from at least two zoonotic events, which seems to me that seems like a bit of a leap for me.
So I guess it's important to think about like these types of as always with phylogeny, right? Like you only have like the present, you only have the samples that you have, and then you're turning them into a big tree. So if you had other samples, like that can change sometimes the way that the tree looks. And a lot of the work a lot of the careful work that scientists have to do is like to make sure that like their data set isn't skewed in some way, right? Like to have some cluster, right, of organisms that would pull the tree in a way that isn't representative of how we think it should be, right? Of everything that's going on.
Yeah,
But assuming that they did their work correctly in that respect, right? Then what they find here is this idea of the two lineages, um, and that would mean that it would have jumped twice, right? That's the implication.
yeah. The idea,
Two spillover events rather than one.
yeah, the idea is that the idea is that they had that there were these two circulating strains were in animals, and then they managed to jump out at kind of the same time, which okay,
Hmm.
it seems like a bit of a stretch, but then I do remember that for like Omicron, which seems to also sprang out of nowhere, came out with three lineages. Uh, and I
Hmm.
remember like when the Delta variant came out, there were like six lineages. So it's not
Right.
unusual, but it
Because it matters how much of that virus is circulating in like the population,
right.
right? So if like using Omicron, I think is a great example because that's one that directly impacted people. So people have this memory, right, of like Omicron, it's come out, right? This new thing that isn't related to anything that it was before. But there's also a few different types of it.
Yeah,
But that's because based on the snapshot that we had of that, we didn't see like we don't have molecular eyes around the world, right? Like surveilling all these different circulating populations. The concept would be that inside whatever population originally gave rise to that lineage, like that was all, there's a whole bunch of circulating strains in that population, and then you're only seeing when it finally like reaches the surveillance techniques. And so that could be the case here, right?
Yeah.
I think is the point that you're trying to make, is that it could be that they were circulating in a bunch of animals beforehand, and there was robust animal to animal transmission, right? Meaning that there were many different variants there with time to differentiate themselves. And it just so happened that that baseline, that base genotype, right, could then emerge twice, right, from that circulating pool. I guess not that base, yeah, that, because the two lineages are related, right, in the fact that they were inside of a circulating pool inside of animals.
Yeah, and also like what, yeah, and because I mean, the original kind of thought was that maybe it was one strain that jumped onto humans, and then you'd see transitional variants, right? And I think they did see some transitional variants, but what these authors say is that those transitional variants are due to contamination or some other kind of like problems, and I feel like they're a bit too glib in erasing those, because I think in one case they said that this could be because they're working on both of these lineages in the same lab, and I felt like that's a bit of a stretch for me.
Yeah, I actually, to me, that actually like sets off kind of just like I don't understand enough about like the actual ways in which they parse their data to make that judgment call. Like their claim on that is something that I'm not good about verifying, right? Like if
same.
Yeah,
I agree. I'm the wrong person to ask about that, but I can sort of see where they might be coming from, but
yeah, for sure. It sounds very reasonable that that could be something that's an issue with the way that data had been analyzed in the past, and that's why we hadn't thought about the possibility of two zoonotic jumps rather than a single one. But yeah, I guess I can.
I think the difference between lineage A and lineage B is actually like, I think only like 2 mutations. So it's not that big a leap to find. If you subscribe to the idea that there were multiple variants that were spreading through animals that we don't see, then I can sort of see that the idea that maybe there was like some kind of event that, but maybe that could have been spread, but I'm not sure. This raises more questions.
I feel like a lot of these, if anyone follows like stories and like ancient evolution and things like that, like it just goes to show how difficult it is sometimes to tease out those stories. And if people have followed ancient evolution stories like throughout their lifetime or whatever, 20 years at least, you'll see how the scientific narrative changes in those stories, because to resolve these types of things in the great past, like it really matters like what data is out there, what new fossils have been found, right? What's the resolution on that? And even an event that is like ostensibly just 2 years ago, like 2.5 years ago, like it's very challenging to be able to definitively say, right, through scientific means that these events happened in this particular way. And maybe that's a little bit of the takeaway from the last 3 papers, is just like that challenge. And we have to also, I think we also have to ask ourselves, like, why these papers? What's the relevance? How are they helping us today to understand things? Maybe they can inform types of surveillance programs. Certainly, they do have like a slant arguing for certain policies in terms of like limiting transfer and how, as we said, there's gaps in those things. Yeah, I think those are my takeaways from all of this. It's kind of like, it's great. I love seeing the elaboration of stories with data, especially ones in the past that might be relevant. I fully accept the limitations on that are quite high in terms of the certainty that we begin to make statements.
Yeah, I think so. And this is the politics is the thing that kind of upsets me about these papers, because essentially both these papers are kind of throwing up middle fingers towards the Chinese government, which might like cause issues during publication, or because the Chinese government aren't the kind of government who like don't come for you if you insult them. So but anyway,
well, yeah.
I'm glad that we don't get YouTube in China, so I don't have to worry too much, I hope. But
but the tone too, I don't know. I mean, this is just a tricky thing. I think we've seen it a lot during the pandemic in terms of literature, right? Like there is a tone sometimes in papers that can be overly aggressive. I mean, these are all preprints,
Yes.
right? These three things. So like they haven't been massaged yet by like editorial teams and peer reviewers, right? Calling out certain elements that might sit less tastefully within those individuals' opinions. So yeah, I guess there's something very raw about reading these initial reports, and as you say, cat fighty in all of it, right? Where it's kind of like this is the expression of the groups and the beliefs of the groups. Like they're using data. Don't get me wrong, right? There's data and analysis done here, but it's also like very unfiltered in terms of like the claims and pointing out like how they feel about what the reality is.
Yeah.
Yeah. It's in all scientific papers, right? It's just that it gets kind of like pulled and diluted as during the publication process and the peer review process.
Yeah, I mean, science isn't a Care Bear tea party. Is like reviews can be mean. Everybody in every echelon can be very. Yeah, it is like a fight. That's why you need data to back yourself up, and that's why. That's why. Yeah.
Okay.
Anyway, I think I've said all I wanted to say about these papers.
Awesome. Then let's push into the fourth paper that we have for people, secondary structural ensembles of the SARS-CoV-2 RNA genome in infected cells. And so this paper is talking about this frame shift element that happens. So we may have briefly touched on this before in the SARS-CoV-2 genome, that their first open reading frame, the piece of RNA that makes the first proteins, it's a big piece, and it encodes the polymerase. That's a really important thing, the RNA-dependent RNA polymerase. But it also encodes a whole bunch of other non-structural proteins. And how do you get that out of a single transcript? Well, viruses have many different ways of doing that. And one of the ways they can do is they can induce frame shift mutations just by like skipping over one base pair. All of a sudden, now you're reading different triplets, different codon triplets, and making different proteins. And so the researchers of this paper use a technique to look at the RNA structure, looking for, I guess, something that is going to induce those frame shift abilities. And yeah, this is a great chart to look at because this is the technique they use. They use a mutagen that mutates unpaired bases. So if there's any secondary structure that exposes loops or there's like some single-stranded areas, those places are going to get mutated. And so then you can deep sequence everything and just by the map of mutations, figure out what that secondary structure looks like.
Yeah. It's fascinating, like how, because sometimes you think of like the genetic code as just like almost a piece of paper, but it's got the structure and function to it that is like it's hard to find an analogy for. I've tried.
Yeah. It's something that we have touched on briefly in some of the other papers that we've covered before, but also on a deep dive, right? Like the RNA structure hairpins. Here's an example of what one might look like. I think this is the one they infer from their mutational studies. Like these structures can interact with the proteins that sit on the RNA and cause them to stall, can recruit new proteins to those sites, right? And all of that is regulatory function. I think the paper that we covered in the deep dive, we were looking at ones where those certain loops could recruit polymerases to them that bypass the regular way of recruiting. I think we've seen on the genetic engineering side on some news episode a couple while back, like engineering these types of structures so that you can control expression. And I mean, that's essentially what viruses have done through evolution, is that they've tried out a bunch of different things. They have a bunch of different variants, and it's been selected for a particular structure that allows the right amount of certain proteins to be made at the right amount at the right time. Yeah, well, I mean that's the big takeaway from this. You can scroll through and see that they really dive into the structure and show that that it is able to create these frame shifts. I think they end up doing like a reporter-based assay, figure 6. They end up doing a reporter-based assay where they essentially set up this system, right? They set up their weird, what they think is the structure that's inducing the frame shifts, and they have one luciferase that's in frame, and then they have one luciferase that's out of frame. And so if the idea is that if some frame shifts are induced, then you will see that the other luciferase make some light, and you can measure the ratio between the two and get a sense of, you know, how much is happening.
Yeah,
how much shifting is happening.
That's a brilliant way of discovering. Also, like a nice little tool to have in the toolbox to figure out. Like, yeah,
and you know, because this is in our favorite pathogen, or I guess like the pathogen on everyone's mind these days, like this type of discovery can be phrased in the way of, well, this is a druggable target, right? Like if we can disrupt this frame shifting ability and force, right, the polymerases to always make the same, the same proteins, they don't get whatever variants are behind whatever other open reading frames are behind the frame shift, then that's a way to disrupt. And with all the caveats, right, that those molecules that might disrupt that may also interact with DNA and RNA in our own regulatory structures. So watching out for, side effects and stuff like that. But, this is one of those druggable targets, I think, is how it gets spun off.
Yeah. Really interesting paper. Really always interesting to find how SARS-CoV-2 works at the genome level.
Yeah. And again, like learning about viruses is, you just learn about regulation at its most basic level. Yeah. So lots of basic knowledge to be extracted here.
Yeah. Speaking of SARS-CoV-2, you've got another paper. So SARS-CoV-2 is associated with changes in brain structure in UK Biobank. So it comes from a UK Biobank where they were running the study that had like a bunch of UK participants between the ages of 51 to 81, and during the study, SARS-CoV-2 happened, and so what they ended up having was like a data set that had people people before and after they were infected with SARS-CoV-2, and this study looks into that and looks into the kinds of changes that we'd see. So they looked into the kind of changes we'd expect to see, and then took a look around for changes that might be interesting things to look at in future. So I mean, the first thing they looked at was, I think, they looked at the olfactory bulb, I believe, was their first one. So I'll just drop down to one of the first figures.
Yeah.
But yeah, so I think they looked at like brain volume as well. They found that like
when they say biobank here, it's like a bank of scans, right? Like
a bank of scans. So it's not like a bank of brain slices, because it's very hard to get your second brain slice after.
Have you ever been consented for taking your brain slice?
I mean, look, I've been on social media, so I feel like I have.
Yeah, and they pair that with the information of whether or not people have been infected by SARS-CoV-2. So then you get the scan paired with the infection status, and they can try to find some associations in this data set.
Yeah, so they tried to make sure that like everything was okay at baseline, because one of the things with these studies is always to make sure that your baseline hasn't got too much variation. There's always going to be variation, but you want to have it in like an area so that you know that what you're finding isn't due to the initial variation. So what you're testing for is what you're testing for, is what we're hoping to find.
And I think brain research in fMRI data specifically, like that's a big thing.
Yeah.
Because like that data is very noisy.
Yeah.
It's looking at blood flow into different parts of the brain during some tasks. Like there's all sorts of variables there that we don't quite understand. So it's good that they've gotten this baseline for us.
Yeah, fMRI, like for reference, like this is for like about 10 years ago, someone made fun of it by basically they used all the standard techniques in the field at the time, and they imaged a dead salmon and used that to prove that dead salmon still had brain function, which actually wasn't so much to to prove that was, but what showed us how ridiculous the analysis strategies were, and how it is easy to find correlation with anything if you don't have the right controls in place for your fMRI study. So
Right.
so since then, there's been a lot of work to try and make things more reproducible for them.
Mm-hmm.
So that is always a concern for studies like this, and
Mm-hmm.
unfortunately, there's been we're not like kind of qualified.
Yeah. But this one's being through peer review, so like we're hoping that like some other experts have weighed in here, saying like some methodological things are going on.
Yeah, I mean, you say that, but I mean, yeah, I'm
Take it
hoping
with a
it's going
grain.
through that.
You always got to take everything with a grain of salt. Like that's the whole point of science
Yeah.
in some ways. Like don't go too far down the committing this knowledge to like your model of the way the world works until like you've been really careful in checking off all your boxes, or you've had some good chats with other experts.
Yeah. So what they found was that there's like changes to the intracranial volume. So I guess it's not so much so I think a lot of that is due to damage to the olfactory sections. But
Mm-hmm.
the interesting thing they find is that the changes to brain function are accentuated as you get older. So so they got these graphs like showing the controls and then the cases in red, and essentially using the ratio of brain to total intracranial volume. So these are mostly to show that there's a difference. It doesn't necessarily show how much of a difference there is.
Mm-hmm.
But yeah, essentially what they seem to show is that as you get older, those changes become much more pronounced. So the idea is that when SARS-CoV-2 attacks the brain, as you get older, your ability to defend against those attacks becomes less good. So
Yep. I mean, and again, that falls in line with a lot of the observations that we've seen already, right? Like on the model side and on autopsy side, right? That there is this effect on the brain, and then combining that with the thought that older individuals are more susceptible. Like here's some evidence now to flesh in a few of those gaps. But not mechanistically, right? Like, yeah, we don't have that sense, but maybe it's like closer to the clinical side. This could have clinical relevance if someone was an expert there to discuss that.
yeah,
I'm not gonna apply that here, but yeah.
I mean, it's interesting for thinking about long COVID and the kind of like ongoing places. So it gives
Right.
people an idea of where to look at. So,
Mm-hmm.
so they looked at like the olfactory bulb, which is like and like kind of the network that's integrated with that, and they looked at various other places. And I think they're building up this idea of like where this damage is happening.
Mm-hmm.
And they were comparing that to influenza, and they found that influenza had its own signature that was distinct from it. So it was yeah
Yeah.
yeah it's an interesting study, again trying to like back up our thoughts on what, because long COVID could be a lot of different things, so trying to isolate it and figure, because I think they try to also pair this with other function tests.
Mm-hmm. I watched a really good YouTube video about SARS-CoV-2 in the brain neurotransmissions. Do you know that channel?
I do not know that channel.
Oh, they had a good one, and they're more on the brain side. They're less infect. They're not infectious diseases. They're also like a much more production valued style video. But if people are interested in learning more about that, they've talked a little bit on their channel about SARS-CoV-2 in the brain, specifically from that like more on the neurology side of things rather than the mechanistic infectious stuff, which I mean we really like.
Yeah, we'd be going into the mechanistic and infectious stuff, but yeah, because we're
But
not
understanding like
neuroscientists.
the relevance to brains is more outside of our wheelhouse.
But yeah, I thought that was an interesting study to bring, so I could say, oh, I don't know about brains, but there's something happening there.
Totally. Yeah. I mean, I know it's nice to see that like there are researchers that are looking at these things, because I mean, I think that's something that it only had a brief moment, I feel like, in the news before it got swept away with the current geopolitical situation.
Mm.
But like that idea that we've had so many infections, and now like we have to address the idea that some percentage of all those people that got infected during the pandemic and are continuing to be infected during the pandemic, right, are going to go on to experience other health complications that we have very little information about. And yeah, so it's good to know that the research is out there and people are thinking about this stuff.
Yeah, and that brings us to the end of our SARS-CoV-2 coverage, and now we're going to look at other cool stuff happening in microbiology.
Absolutely. And the first one is remodeling Yersinia pseudotuberculosis to generate a highly immunogenic outer membrane vesicle vaccine against pneumonic plague. So to understand this paper, we have to understand Yersinia pestis is the causative agent of pneumonic plague, and Yersinia pseudotuberculosis is a related foodborne pathogen. I think it's a foodborne pathogen, or enterocolitica is also one. Anyways, there's a bunch of Yersinia that cause some other diseases that aren't as bad as swollen lymph nodes and wasting away lung disease. And actually, it was previously found that the extracellular vesicles of Yersinia pestis do have like some of the molecules that we would want to elicit immunity towards, and they had used that in a vaccine scheme, but it didn't make enough of the antigenic molecules that they wanted. Stuff involved in like toxin secretion and stuff like that. So the researchers of this paper turned their eyes to a different Yersinia species that maybe is more amenable to making these things in some ways, and that's where the story begins. Outer membrane vesicles we've briefly talked about before. I don't think we've covered a deep dive on them yet.
Yeah, I don't think so. But they're basically like
pending.
They're virulence factors, right? So like during an infection, Yersinia pestis will produce a bunch of these that will have various like molecules and proteins on them that will really mess the immune system up. And so yeah, they're quite important for infection.
Yeah, they bleb off the surface. I think I mean, I didn't when I was first learning disease stuff, I wouldn't have called them like important for infection per se. I think they were just, I thought they were just weird curiosities at the time. But then if you think about it, like having those virulence factors out on membranes floating around, like of course it's going to elicit immune responses and potentially push the disease state in one direction or another.
Yeah.
So they use ones from like a less virulent strain, right? Pseudotuberculosis here, and they immunize mice with them.
Yeah, Yersinia pseudotuberculosis is thought to be the direct evolutionary ancestor of Yersinia pestis, because the two species share about 95% of their genetic identity, and they have like common like plasmids. But I think like Yersinia pestis has like had a few gene deletions and a few additional plasmids, but it's kind of unstable to work with, so they'd want to work with something like Yersinia pseudotuberculosis.
Yeah, and again, I think they hinge on that idea that the outer membrane vesicles from pseudotuberculosis, like they're just able to make more robust ones that have the right antigens in them. So it's really just in some ways luck of the draw in that sense, that like, oh, here's something related that essentially makes very similar things that we can use as a immunogen in a vaccine, and it's also its own adjuvant.
Yes.
Probably because it comes with all that membrane bits as well, right? That it's triggering other signals in the immune system to say like, oh, pay attention to the stuff that's associated.
Yeah, I think that's a really good way to approach like fighting off bacteria. Target the virulence factors and the toxins that are causing the issues, and then that will allow the immune system to respond to the bacteria directly. And they do test that in mice. So they find that the mice are safe.
Yeah, it works, and it does increase the antibodies against this LCRV, which is the immunogen that they're interested in. It's previously been used in protein-based vaccines. So, they have this sort of line of thought that they know that this is one of the targets that can help produce productive immunity.
Okay. Yeah. They do I think it's quite important to like test the immune response to try and figure out how something is working, because
Mhm.
when you're working with an animal, you want to make sure it's relevant for
Yeah.
humans, so they kind of do that here.
Mhm.
Um,
Yeah, they
but
show
yeah, very
the
promising.
expansion of the T cells that are relevant for this. So you see it both on the side of it reduces disease in the model, but it also has the immune correlates that then you can go say, okay, this might make it a good, this gives you a sense of what that mechanism is, and then you could go on and, expand that study. So there are lots of, there are a bunch of plague vaccines out there. Finding new ones, I mean, this is a kind of crazy thing to think about, but finding new ones seems to be, at least in the US, the domain of like biodefense sort of research. Like the fear of plague being used as a weapon drives a lot of these questions. But it's also important to think that plague is also still endemic. In the States, it's all to the west in groundhogs, so it's possible to get plague to spring up there, and in China, a lot of groundhogs there have pestis in them.
Okay, I'm going to avoid all groundhogs forever now.
Yes, yeah. Groundhogs are the considered the reservoir, I think, for these things, and then fleas, right, are the vector.
All right, so avoid groundhogs, fleas, and that covers Bill Murray and the Red Hot Chili Peppers. So need to.
All right, that's all I wanted to say about this one. It's a kind of an interesting new vaccine approach. Maybe not so totally new, but newish.
Yeah. So bacteria eat nanoprobes for aggregation-enhanced imaging and killing diverse microorganisms. Now, the original title of this paper was Novel Pac-man Imaging and Therapy Strategy for aggregation-enhanced imaging and killing of microorganisms. So the original title of this was Pac-Man, but I feel like it got chewed up during review, and I feel like quite badly about that, because the original so actually the original title was Pac-Man Bacteria Eat Nanoprobes for Aggregation-Enhanced Imaging and Killing. And yeah, so the original preprint is really wild to read, but
has a lot more personality than this published version.
Yeah, it was a lot more like punk rock, but now it's much more official.
But the basis of this technology is tiny gold nanoparticles covered in some sugar get passively absorbed by bacteria through their, I think it's ABC transport. So that's just like the way in which bacteria get sugars into themselves. These gold particles can hitch a ride on that system and accumulate inside of bacteria.
Yeah, so these like bacteria usually eat glucose, and they've got these glucose particles that are attached to it. So when the bacteria thinks it's eating glucose, it's actually eating this gold nanoparticle, like Pac-Man, our round friend who can't control himself.
Yes. Well, I feel like the Pac-Man thing is good because it's like those little dots, right? He's eating little dots everywhere, and these are just tiny dots essentially that get into the bacteria. And then through using light, they're able to cross-link these pieces of these gold nanoparticles into larger particles that then can be detected through other methods.
Photoacoustic imaging, which is they have a laser that hits these nanoparticles and causes them to produce ultrasound, which can be detected using an ultrasound detector. So using light to create sound, which is again really interesting and cool.
Totally. I've never heard of this particular way of imaging before. I have heard about ultrasound, and that there's, I mean, this is like a bit of a tangent, but I've heard that there are some genetic modules that you can engineer in bacteria to make them more amenable to ultrasound detection.
Oh, yeah, I think you can do these things like microbubbles were a big thing about 10 years ago, where you use those as contrast agents.
But here the agent is these gold particles, and they're not being excited by they're not detectable per se by ultrasound by itself. It's that they're induced to create ultrasound. So strange.
And you can also sorry, I missed that last bit.
Just physics is a strange beast.
Yeah, it really is. And as you can sort of imagine, like that can also turn into heat transfer, or so you can actually turn these into a kind of therapy against the bacteria
Mhm.
because you can make them heat up, you can make them produce alpha, so you can image them and kill them at the same time.
Right. And so in this paper, they go through a whole bunch of different case studies, essentially showing in a wide range of bacteria uh, that these particles get taken up, and then showing that they can then image those bacteria in the context of like a mouse flank or tumor.
Yeah, so we've got some nice images of, so firstly of them aggregating after being exposed to being irradiated with a laser.
Mhm.
Uh, and then we've got some images of them inside mice. So they've tagged these with a fluorescent marker, so you can see these images of the whole mice. But then they've got these ultrasound images, which kind of give a lot more resolution on what's happening on inside the specific organ, so you can get a lot more detail of where the bacteria are and where they've spread. So that's actually quite useful for understanding how back.
Uh-huh. Yeah, I was struggling between choosing this paper and another paper I saw about, last week we talked about the pet
Yes.
tracer that was being used inside macaques, and then I saw a paper that they used the pet tracer in HIV patients to find HIV. But then I was like, actually, no, like we already talked about that one. Let's look at other imaging things. But the same sort of excitement for these techniques applies, right? Being able to in vivo look at where certain organisms are going, right, gives us this insight onto the disease process that we wouldn't have otherwise had, that we would lose because we would kill organisms and slice them or homogenize the whole thing and get the average.
Yeah, I mean, really fascinating. I mean, I'm not used to interpreting ultrasound, but I think that there is
Mm.
quite a quite a lot of cool things here. I think I only tried ultrasound on a mouse once, but the ultrasound detector was larger than the mouse, so it wasn't really great. I think you can get much better technology now these days. Um,
Uh-huh.
but
So that's the imaging part, right? They put it in the context of mouse, also a mouse tumor as they look at,
yeah,
and also mouse gut. So they can see basically these bacteria congregate wherever they inject them, and then and then the final part of this is showing that they can do that other function where they then like vibrate the particles so hard that I guess that's that's
yeah,
my assumption of what's happening. Yeah, vibrate the particles so hard that it causes heat to build up and destroy the bacteria.
yeah, and you kind of see this with like they got these like electron microscopes of these normal bacteria, and then they kind of these like really broken up versions of the bacteria, which are
Yeah,
quite fun.
that's the scanners. That's the, the thing from scanners with the head
Yeah,
exploding.
yeah, that's
That's
right.
that's essentially what we're seeing here, but for bacteria in still images.
Yeah, completely. It's mind blowing. Um,
um,
so
yeah,
yeah,
and then they use it again in an animal model to show that they can kill some antibiotic resistant bacteria.
so
This is like this is staph abscess. This is a model that I saw a lot of in the lab.
oh yeah, oh it's
Back
not
in the day.
great, it's not a pleasant model to work with.
No,
But yeah,
no, no.
um, oh yeah, I
Okay.
think that this is an interesting approach to antibiotics as well. Just like using this Trojan horse to get this molecule in that isn't actually immediately dangerous for the bacteria. So
Mhm.
that means that bacterial resistance isn't going to be as much of a problem because I think
Oh
it only
yeah.
because outside of the treatment, there is actually no reason for the bacteria to evolve resistance to it. But if this
Right.
leaks out into the environment, the only
I,
concern is, sorry,
oh no, I just want to say you're going to say the only concern. I just wanted to link it back to you had picked out a paper I think about SARS-CoV-2 and like these hairy balls of some
yes, that's
sort,
right.
nano balls. It's the same thing, right? On the safety side, like what do they do to not the other parts of the body?
Yeah, like do they like because I think they say that it does end up in like say like in the places that are used to clean blood, so spleen and liver. But the question is whether they cause damage. I don't think they find that they cause much damage, but this is very early stages, and I think there's quite a lot of concern about that for nanoparticles.
Right.
So it'll So more research is needed, obviously. I mean, I guess we're scientists. We always say that. Like,
Yeah, that's just the everything line.
yeah, more research is needed. Give us more money. And then you want answers? Give us more money.
All right, moving on. We've got molecular and cellular dynamics of early embryonic cell divisions in Volvox carteri.
Yeah, this
Carteri?
Volvox is an interesting organism because it is a microbe, but it's like a colony microbe. So
Mm.
it is multicellular, and then you have like, so it's kind of like an algae, kind of like a plant, but it's got these it it's segregated in a strange way. So you've got like these gonidia inside of it as well, which
There's
are
division of labor, right? I think
yeah,
that's one of the fascinating things. It's colonial in that these cells are all related to each other. They've all like just divided from the same cell, something like this. But then there's this division of labor that happens where the somatic cells on the outside have cilia that allow for motion, but the sex cells on the inside, they're just like huge things just responsible for cell division and giving rise to progeny.
yeah. I think the question this paper wanted to ask is like, how does cell division actually work in algae? Because apparently there isn't as much information out there,
Mm.
and they're looking for like a good model organism. They decided to on this one, but I think due to ease of study.
The,
Um,
the size of those cells, like
yeah,
just
look
see
at
how big they are.
yeah, it's
And then it
it's a
also,
really like
oh, I just want to say they used it. They also seems to have used it because they can get chlorophyll. Right, this is an algae. They get good contrast to see some of these
yeah,
events. Yeah.
that's right. So there's a couple of images in this paper, and what they did was they took a bunch of different proteins that are involved in cell division and tagged them with yellow fluorescent protein. And
Mm.
because of that, they're able to look at what each protein is doing during cell division and like how they're dividing. So we've got some cool little images. Sorry, not images, videos. And we've got some really amazing images showing like there's all these microtubules that link onto the DNA as it's dividing and pull away the individual chromosomes. So we get some like really cool images of that. But I'm here for the videos, which are down here somewhere. Uh,
This is I feel like this is a lot like just when we learn about cell division in school. This is like a very early, right, like science lesson that people learn, but it happens slightly differently in different types of cells. So getting that knowledge for algae is part of learning about algae.
yeah, I mean, I think because plants have this thing where they produce these things called a phycophore, whereas in eukaryotes they have these centrioles that coordinate it. And I think what they find here is that there's a bit of both systems at work here, which is a new finding.
Are you gonna play it again for us, or oh, there's
There's
many videos.
a couple of other videos. So this first video is just uh, tagging everything in the cell. So this is the idea of like showing the whole nucleus and watching everything divide in it.
Mm-hmm.
Um, and then you see it turning into two nuclei. So the idea, the nuclear membrane kind of,
Excludes the dye or
yeah.
the fluorescent proteins.
And then we've got the second one, which looks at chromatin, which is the thing that ties up DNA. So what we can see is like the chromatin winding the DNA up, and like so it can, and then pulling it apart. So there's making sure that each daughter cell has the right level of DNA. And
That's a great video.
Yeah. So
I feel
you've
like
got
also the early information that we get as like students about cell division, onion cells are often looked at, right? Like all the slides people have are like stained onion cells for anaphase, metaphase, and all that type of stuff. Now you can take videos. I'm just like from the technological side, like now you just like make transgenic. Now you make transgenic whatever you're studying and take videos of it under fluorescent microscopes.
I mean, that's also another great thing about having an alga like this is that it can operate on like that single cellular level, whereas with an onion, you might kill the onion cells through extracting them, so it might be quite hard to find one and watch it through cell division.
For sure. Also, it's hard to make a transgenic onion. It's easier to make a transgenic algal cell.
Yeah.
I assume. That's also
I
that's
haven't
actually a
tried
big
to line.
assumption, but yeah, I assume.
So yeah, this is looking at the microtubules whose job is to move all those globs of DNA we saw in the previous image.
Mm,
And yeah, you can sort of see. So those are the three videos. Um, but yeah, I thought it was
nice.
a really cool way to show this basic process that we were taught it at school, but we didn't necessarily like see all of it. And it's like, so I think this should be a good way to show these. I'm sorry, my brain is fried a little bit.
I'll also say what I like about this is I just like bringing more attention to interesting model systems, right? Like Volvox is interesting not just for that's giant gonitophore that you can visualize cell division on, but you can also study all this interesting colonial stuff. I mean, that's one of my like fascinations in science, right? Like how do you like unicellular, how do single-celled organisms communicate with each other and create more complex structures. Like Volvox is like a really cool model from that perspective because it's like making these these colonial living spaces. So yeah, that's maybe one of the other things that I take out from this, just excitement about about different model systems.
Yeah, I completely agree. Speaking of cool model systems, next paper
Yes, innate
is
immune pathways act synergistically to constrain RNA virus evolution in Drosophila melanogaster. So, we've been talking a lot about trying to predict the evolution of SARS-CoV-2 in different cases. But of course, like that model system, SARS-CoV-2 in a human is not something that we do reproducible experiments on. But there are many pairwise interactions that exist out there in nature, and some of them we have a lot of fine-tuned control over. Drosophila is one of those systems that we have just like so much control over in terms of making mutants. I think previously we had seen people can do like crazy behavioral stuff with Drosophila as well.
hmm,
In this case, they have a bunch of Drosophila and they give them specific mutations in their innate immune system. And then they have a Drosophila virus that they passage within those poor Drosophila that are deficient in different ways, and they look at the viral diversity between passages. So they have 10 passages that gives them lots of snapshots into the different mutations that might occur, and they set out to show how the innate immune system is shaping viral evolution, which of course makes a lot of sense because the immune system is trying to kill viruses, so viruses need to escape it. If that pressure is removed, then we may see, a different population structure emerge from the populations of viruses that are growing in those deficient organisms.
yeah, it's really fascinating where they delete like different parts of the innate immune system. So the parts of the innate immune system that are designed to attack viruses, there are parts that are designed to attack bacteria. And one of the
Mm-hmm.
cool little things they found here was that even if they deleted a part of the immune system they didn't think was relevant for viruses, that still caused a change in viral diversity, which
Yes.
which kind of indicates that it's
Uh,
like an interconnected system, right? That there's a lot of
totally.
things going on that we don't necessarily know, and that's really interesting.
Yeah, and I think this is, I think, the chart that says it all essentially, because these are charts of mutation abundance. So you get these different colors, the size in which they take up that entire bar. That's how much prevalence, I guess, of these different mutations are, and then you have time going from left to right. And these are two replicates. So it's two biological replicates. They have some initial set of viruses introduced at the beginning. They sample them every generation, and then each of the bars going top to bottom is just a different mutant. And so you can see at wild type, the viruses don't really have much space to change things up. They end up, maybe there are some genotypes that emerge, but then they become successful and they fill the niche, right? This is the only genotype that survives in these flies. But in all the subsequent places where they have different mutations, you can see the type of diversity that comes out. I guess there are some that don't. I mean, what's interesting too is some of them they just like totally different. You don't get the same mutations either, right? Because it's really just the removal of a selection pressure means that other maybe neutral selection type things could be happening, or stuff that we don't even understand in terms of like how a virus might find a more efficient way to replicate. Those become viable options, and the viruses just go wild. But yeah, I think the point that you brought up is a really great one, that there's an interconnected nature as well between these systems, and they work to create a very specific evolutionary environment inside of our bodies that viruses are contending with, and that are evolved to find a path through in many ways.
Yeah, a fascinating look. I think I've said all I can say about this one.
Yeah. So moving to our last paper then for today is
a comprehensive phenotypic screening strategy to identify modulators of cargo translocation by bacteria type 4B secretion system.
I think to me this is like really going back to some of the techniques that I saw a lot of when I was going through grad school, in terms of, you know, we have bacteria, we have many different compounds that we want to test in them. One of the great things about doing bacteria is you can just like replicate the same experiment in a tiny well of a microliter plate, and you can get you can basically test the same condition over and over again with just like one little change. And so the model system here being used is Legionella, which is a pathogen that has a secretion system. It secretes a whole bunch of different effectors into cells. And then they have macrophage cell lines, the raw cell line. I think that comes from mice. And they mix them together. And in that system, the Legionella are going to inject their effectors into the macrophages, and a dye is added so that if the certain types of effectors get in, then the dye changes color. And on the basis of that assay, they then, in every different well, insert some random molecule that could be a drug, and see what stops the injection process from happening.
Yeah, it's really, so they kind of saturate both these molecules with this thing called CC4AM, which is like a fret molecule. So it's got two kind of fluorescent tags that quench each other when they appear green when they're together. And then they have like this gene, I think it's beta lactamase that can be transferred between the cells. And if beta lactamase is present, then that can cut the molecule up and cause it to turn blue. So when we see these cells, if they turn blue, then that means that these type 4 secretion systems are working. But if they stay green, then it means that they're not.
Right. Oh yeah. That's a good point. I should add that like the thing that these Legionella are injecting are something that we engineered into them so that we could basically so that we could detect whether or not the injection is happening. So the beta lactamase is fused to like one of the secretion signals, right, that Legionella uses. And yeah, they're looking for inhibitors, or I guess they can even find in some ways activators if they see things that I guess light up earlier, something like that. Yeah, of this secretion system. And it's just a big screen in a way. And you can see in figure 2 the type of workflow that gets used on assays like this, because the primary screen where they're just looking at which molecules are disrupting things, they have to be whittled down in order to find candidates that would be useful to study for other reasons. So, you know, they have to confirm things that, you know, sometimes something happens in a well, but that was just like a random event. It didn't actually cleave the molecule in the way that they thought they did. And so those get like followed up on. That like removes a whole bunch of hits. Then they do other types of assays in order to sort of narrow down the molecules that are actually having this effect of preventing secretion.
Yeah, it's this kind of technology which reminds me of like throwing spaghetti against the wall to see what sticks. And sometimes like you get spaghetti that's you hit a particularly sticky part of the wall. I don't want to. This isn't a comment on the state of my kitchen, by the way. But yeah, So they need to like make sure, in order to figure out whether they're done or not, they need to do multiple checks just to make sure.
So, but that's one of the pleasures, I think, and the power of working with bacteria is that because you can set them up in these small little assays, like you can do this type of like spaghetti throwing experiment where you get closer and closer to just the stickiest stuff. The stuff that makes the biggest mess. I don't know why we're trying to find the biggest mess, but like that's what we'll be doing.
Apologies, my second screen is about to shut down and I can't do anything about it until I find my remote control. So ignore me, I'm just going to be rummaging around.
Or we can just wrap it up because that's the last paper that we got.
Oh yeah, of course, we can just wrap it up. So that makes it a show.
Yeah, so join us next week for more microbiology news.
Yeah, and let me think. Apologies, I've been very sick. So if you feel like after watching this video you're getting flu-like symptoms, and I'm very sorry. I'm not kidding. You can't actually get the flu from a YouTube video. I need to clarify that. But anyway, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything correct. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yeah, totally agree. We both believe that peer review is this process that we can all participate in as long as we're willing to dig into that literature. If you enjoyed listening to us ramble on about microbiology today, then let us know. If you think you have something to add or found something unclear, please use the comments.
It's been a pleasure chatting with you, Danny.
Same here, Faz.
Tune in next week for more microbiology content. Bye-bye.